Good evening, everyone. I'm just going to open a poll as usual, just to check whether you can hear me. So if you can click, that's perfect. Thank you. Okay, so we are on lecture 5 today, and we're going to be talking about capillary electrophoresis. So we can just go straight in. So our learning objectives for today is to discuss the principles of capillary electrophoresis, to describe the main components of a typical capillary electrophoresis instrument. Then we're going to talk about different separations modes that we can utilize to analyze our product, and then discuss the application of CE in biopharma in particular. Okay, so we are still here on the kind of second row on our table. So talking about electrophoresis. So our last lecture 2 weeks ago was about slab gel electrophoresis, which is a slightly older technique. And today we're going to be talking about a bit, let's say, newer technique that is used for electrophoresis, that gives us information about purity and integrity, about molecular weight of our sample, isoforms, and it's used in all stages of development. Okay, so as I said, we were talking about classical electrophoresis. So if you remember that lecture from 2 weeks ago, there are some advantages, but many more disadvantages of um, this kind of analysis. So classical electrophoresis is relatively inexpensive, and it allows you parallel operation. So that means that if you remember, you have your gel, you have little wells in your gel, so you can load different amount of you can load few samples at the same time, depending on how many wells you have. So you load few samples and you are actually letting them run and separate at the same time. So everything is happening at the same time. So all of your samples are going through the gel at the same time. So that would be my parallel operation. But when it comes to these advantages, there is a lot, as I said. So it's quite a manual technique, as you will see when you come to NIBRT and do your practicals if you haven't done SDS stage before. So operators really needs to do a lot, needs to be there, needs to set up. So there's a lot of input from operator, which can be really time consuming and laborious. It adds to long time, so it takes a long time until you actually get that final result. Efficiency is really low, and it's really difficult to detect what you have on a gel. So whatever you have on a gel, if you remember, you have to stain, and then staining when you're looking at the dye, that's not really quantitative method, so it's only like semi-quantitative method for your samples, and you can't really automate much because, as I said, you need a lot of input from an operator when using this technique. Capillary electrophoresis is similar to slab gel electrophoresis, but it's not done on a slab gel. It's done in a small capillary. So you can see here we have a capillary of a different length with really small diameter, and you can do your electrophoresis inside that capillary instead of a large slab gel. It also provides you information about size, charge, identity, and purity, same as electrophoresis, but it can be used in real diverse application range. So, of course, we are interested in using it in proteins, for peptides, but it can be used for amino acids, looking at nucleic acids such as RNA and DNA, and you can even use it to analyze whole cells, so really wide application. So what is actually CE and what are the principles? So because it is electrophoresis, the main principle is the same as it is in slab gel, just electrophoresis. So that's main driving force of separation on a thin capillary. So the main driving force is the separation of charged samples because of the influence of electric field. So we still have our capillary that will come, where our sample will come through the capillary and separate based on their charge, because we have our two electrodes. So we have our negatively charged electrode, we have our positively charged electrode, we have high voltage supply, and we have a buffer that helps us conduct that electric energy. But in CE, we have additional force that helps separation. So we don't have just electroforce. We don't just have the electrophoretic force. We have also a phenomenon that is known as electroosmosis or electroosmotic flow, or EOF. And this happens, so this additional force that separates samples in capillary electrophoresis happens because of the small diameter of the capillary and actually the material of which capillary is made. So this contributes to this another aspect of separation process. So actually what is happening inside your capillary, so if you were to cut your capillary and then look through it, you would see that the inside of the capillary capillary is made out of fused silica, and inside of capillary is negatively charged at pH higher than 3. And that means inside is negatively charged because there are a lot of silanol groups inside our capillary, and they are negatively charged, so they are deprotonated at pH higher than 3. Okay? Because of that negative charge, when we put a buffer inside our capillary, this negatively charged walls will attract positively charged ions or cations from the buffer, and they will actually create an electric layer. So actually the way it looks like, so the first layer of these cations that will be really attracted to negatively charged silanol groups form a really strong fixed layer. Okay, so that's layer that's really fixed to the sides of the capillary, but this layer is not sufficient to totally neutralize all the negative charges that we have on our silanol groups. So that's why another layer of cations forms close to the edge. So this layer now of cations is not as fixed to the side as the fixed layer is, and this layer we call mobile layer. So that mobile layer then neutralizes negative charge, and then the layer above that one is just the layer of your bulk solution of your other anions and cations. So your fixed layer of tightly adsorbed cations and then your mobile layer, which is rich in cations, they form this double layer. So double layer that is really rich with cations. And because this double layer is really rich with cations, all of those cations are moving, so remember this is a mobile phase, they are moving towards the negatively charged electrodes or cathodes. And because they are moving, they act almost like a river or like a train that carries everything else towards the cathode. So this is electroosmotic flow. It's just a force that moves everything towards the cathode because of this double layer that is being formed. So it doesn't matter what you have here, whether you have neutral ions or you have charged ions, they will all move towards cathode because of electroosmotic flow. And usually electroosmotic flow is beneficial, so it helps us even a bit more to separate our samples, but sometimes needs to be controlled. So there are some cases in which we don't really want that electroosmotic flow. So for example, if we are operating at really high pH, that EOF can be really, really rapid, resulting in elution of solutes before separation occurs. So at really high pH, we have really a lot of negatively charged silanol groups. That means that we have that really thick double layer that will just carry everything towards the cathode so quickly that nothing will actually be able to separate. Opposite thing is if we are operating our analysis at really low pH or moderate pH. In this case, our silanol groups will be protonated, so they're not going to be so negatively charged. So it can then mean that nothing will actually move, everything will move so slowly through your cathode that actually ions can start absorbing to your capillary surface, which is also not good. And then some of the separation methods actually need absence of EOF, and we need to reduce or completely neutralize EOF so they don't interfere with our separation mode. So we'll mention that later, but things like isoelectric focusing on a capillary or capillary gel electrophoresis or SDS page basically in a capillary, they require reduction or absence of EOF. And we can control EOF in many different ways. So for example, we can control EOF by controlling the strength of electric field, which is proportional to change in EOF. So the higher the strength of electric field, everything will be moving quicker. This is easy to control, but it can lead to Joule heating, which means that, increasing electric field just means that we will increase heating of everything that's inside the capillary, because the higher electric field, the higher energy that is being released. So that can actually damage our sample. So we can control EOF in other ways. We can control it by our buffer pH. So we can decrease or increase our pH of our buffer, and then we will either decrease or increase EOF. So at low pH we will decrease EOF, and high pH will increase EOF. Then we can also use some kind of modifiers or surfactants or polymers or coatings. So anything that will kind of neutralize and block those silanol groups that can help us to control EOF, or even temperature, which is easy to control. So increasing temperature, for example, changes viscosity of solution, so decreases viscosity of solution, and then that helps with increasing EOF. Okay? So you can pick how to control it depending on the analysis, depending on the method that you are utilizing. Unique feature of EOF in the capillary that it gives us this fat flow velocity profile. So if you are using something like HPLC, which we're going to talk about at the end of this semester, so basically liquid chromatography, you have a pump that is moving liquid through a small column. And every time when you have a pump, that generates this kind of laminar flow of sample through your column. That means that there is quite a lot of shear forces at the wall that are stopping your sample from moving completely kind of uniformly through the capillary. So you can see this main part is moving quicker than these parts that are close to the sides that are being kind of held back because of shear forces. In our EOF, in our capillary electrophoresis, we don't have a pump that's moving things through. We have electric field that is moving things through, and the electric field is acting the same on the whole sample. So we don't have those shear forces are not as strong as in something like UPLC. So as our sample is going through, it's not really spreading into this laminar flow. We have this flat flow of sample through the capillary. That means that sample will go through our detector in a really flat flow as well. So we're going to get much sharper peaks at the end. So this leads to really high resolution. So capillary electrophoresis can actually really lead to high resolution. You can be really efficient to separate different samples. Okay. So next few slides are about the main components of the typical CE instrument. So of course you need a high voltage supply, and actually this voltage supply that's supplying your capillary electrophoresis and moving everything through is much higher, much stronger than the one that you're using for your gel electrophoresis. So for gel electrophoresis, we're talking about, let's say, 130 volts, while here we are talking about 15 to 30 kilovolts. So we can actually operate using much higher electric field, which means that we can separate everything much quicker. Then after that you need a capillary, so your sample is separated inside the capillary. So this capillary can be of different kind of diameter, and it can be of different length. So those diameter of your capillary can vary between 25 to 100 micrometers, depending again on the analysis that you are doing, and it can be really long, you can buy it up to 1 meter long, and you can cut it as you need to, okay, for your analysis. And the reason why we can operate under such high electric field when we are using capillary electrophoresis is because this capillary is really long but really thin. So we have large surface area to volume ratio, which enhances efficiency of heat dissipation. So we have quite a large surface here, which means that heat that is being generated because of the high electric field, it's actually going to be dissipated because of that high surface. So we can really use higher densities, which current density, which speeds up the rate of migration. This capillary is kind of brown in color, so you can see these parts here. This is because most of the capillary has this polyamide coating that allows more robustness for a capillary, so it does not break. But there is a part on the capillary that's missing polyamide coating, and this part is called the detector. Okay, or sorry, detection window. So this is a window that gets aligned with your detector on your system and allows you to read the signal from your capillary. So if you look at this back here, so what you have your capillary in your reservoir, so in kind of like put into your buffer reservoir, your sample reservoir. So your samples go through the capillary and get separated until they reach detector. So this is where we have a detector window on the capillary. They get detected here, and then they come out and go to waste, or they are, or we can collect them if we want for next analysis. Okay, so capillary needs to be put in a cartridge like this, so it's fed through a cartridge to prepare it for use. So you can have different length loops attached to allow you different lengths of capillaries to be used. So this is quite a short one, so allows you just to put in a shorter capillary, but you can have really like looped and long ones in case you want your capillary to be longer. And sometimes you want to allow more time for your sample inside the capillary because that helps with separation. So you can change the loop and actually put in a longer capillary. And this is a window, so that's the part of your capillary where you don't have polyamide coating, and that's aligned with the detector and allows your detector access to your capillary window, so you can actually detect whatever is coming off from that capillary. Then, of course, the detector, so detector gets aligned to the detector window on your capillary, and your detector can be, those detectors can be few different ones. So here we have an instrument, Beckman Coulter instrument PA800. So this is the most popular instrument that is used in QC as a capillary electrophoresis instrument. And detectors that come with it, that you can get with it, can be UV detector, they can be diode array detector, or DAD, or laser-induced fluorescence, or DAD, or LIF. DAD and PDA, they would be the same thing, so they're kind of diode array detectors. And you pick them depending again on the analysis that you are doing, and their sensitivity is slightly different. So if you want to compare them to slab gel electrophoresis that we were talking about, your UVA and your DAD or PDA would be something similar to Comassi staining, and then more sensitive. And sensitivity of laser-induced fluorescence would be more kind of similar to silver staining, so it's a bit more sensitive. So UV detector would be the most widely used detector. So you basically have your lamp that emits light in the UV spectrum, so below 200 nanometers up to visible spectrum, and you pick your wavelength that you want to use, and usually that's the wavelength of 280 nanometers because that's the wavelength at which our proteins are absorbing light, if you remember, because of those amino acids, tyrosine and tryptophan mostly. So with your monochromator, you pick the wavelength that you want to use, so that wavelength is shown on your sample. Then your sample absorbs light, some of the light is transmitted. Transmitted light is detected by a photodetector, and then we get a digital signal. So we get peaks as our samples are going through the column, and we can look at those peaks and analyze them. Then you can have diode array detector. So this allows you to scan across all wavelengths at the same time. So you don't have to pick just one absorbance, like with UV detector. With UV detector, you can only pick one absorbance, one wavelength. This allows you to actually scan your sample across all wavelengths, and then you can look at all of those wavelengths during a run, and then and analyze them after the run. And this is really useful for method development. For example, when you don't really know what would be the optimal wavelength for your sample. Okay, so when you're still trying to figure it out, you scan over all the wavelengths and you see where your sample is absorbing the best. And it's also good for confirmatory analysis to confirm that something is of good purity, or to confirm identity, because you can scan across all the wavelengths. You kind of get like almost like a fingerprint, and then you can compare that with your reference standard. The only thing you have to be careful with is, because in your lamp you're not picking just one wavelength, all of them are there. So that means all the energy of your lamp is focused on a very small region of the capillary. So that can really damage your capillary, and it can damage your buffer. So you have to make sure that you have coatings that are compatible to use with it, and buffers that will not decompose under these conditions. And then you can have LIF or LIF detectors, or laser-induced fluorescence detector. So basically what you have is a laser that will emit certain wavelength, and that will excite certain fluorochromes and then give you an emission. So this is used for analysis of compounds that either naturally fluoresce or that can be fluorescently labeled. So things like carbohydrates, nucleic acids. So you can actually add a dye, fluorescent dye, to them, and then you can use this method to detect them. And LIF is really selective, really highly sensitive, and typically yields 10 to 1,000 times better sensitivity than UV absorbance. And you can pick a range of different reagents from the market that allow you to add a fluorescent molecule to a specific group of your analyte, so you make that analyte fluorescent, and then that way you can detect and you can see it. So LIF mostly at the moment is used in biopharma to detect glycans, so to detect sugars on our antibodies. Then you have your buffer and your sample reservoirs. So you basically, capillary, once it's put into the system, capillary doesn't move, but you have like little stations with your inlet buffers and your outlet buffers, and another little station with your samples that are moving towards the capillary as you need them. Okay, so this reservoir can be inlet, or it can be it can be changed for your sample depending on whether you are just injecting a sample or you are injecting your buffer and running the method. And then you have your outlet reservoir. Okay. There are a few ways in which we can inject our sample onto a capillary. There is an electrokinetic way. So this means that our capillary inlet is inserted into the sample, and outlet is inserted into a buffer vial, and then we briefly apply voltage. And once we apply voltage, sample is just drawn into the capillary through a combination of electrophoresis and electroosmotic flow. So if we have sample here, we apply voltage, our sample just gets drawn into the capillary because it's actually attracted to negatively charged cathode. This is not ideal method, because you can imagine that the components that migrate more rapidly in the electrical field will be overrepresented in the sample compared to slower moving components. So once you apply voltage, of course, if you have cathode on the other side, everything that's positively charged will jump from the sample into the capillary much more readily, much quicker than anything that's, for example, negatively charged. So you can really overrepresent positively charged, for example, in this case, and get false results. Another way in which you can inject your sample is hydrodynamic way, and this is by pressurizing the sample to push it into the capillary, or creating a vacuum at the reservoir to pull the sample into the capillary. So you can either pressurize this sample reservoir, so you can push a bit of sample into the capillary, or you can pull a vacuum on your outlet reservoir, So you can actually pull your sample into the capillary. And your injection volume can be controlled by either adjusting delivery pressure or delivery time. So usually lower pressure is a bit better, so a bit more gentle for your sample. So um, you are just setting the delivery time, so how long you will be applying a pressure or a vacuum. And you have to just really control temperature in this case, because temperature will affect viscosity. Okay, so that will then affect the reproducibility of injections in both cases. So I see Rita, you raised hand. So if you have any questions, just feel free to type them into the chat bar. No, that was a mistake. Okay, so we'll just continue. So your CE results will be shown as electropherogram. An electropherogram is quite similar to chromatogram. You will see when we start discussing liquid chromatography, high performance liquid chromatography. So you have on your x-axis, you usually have time, and then on your y-axis, you have detector response. So that might be UV response or maybe response from your laser-induced fluorescence. And then you get your peaks as your sample is going through a detector. You can see we get pretty sharp peaks because of the flat velocity, flat flow velocity. And what we are measuring is migration time of samples through the capillary. So for this sample, migration time, for example, would be 4 minutes. For this sample, migration time would be 10 minutes, and so on and so forth. So that would be kind of a qualitative attribute of our sample parts. So how quickly they migrate through the capillary to the detector. Okay, so what I'm going to mention now in the next few slides are different separation modes that we can use when we are using CE analysis. So several techniques can be used. So similar like with slab gel, when we were saying that we can use SDS, that we can use native, that we can use isoelectric focusing and 2D gels. Here for CE we can also use capillary zone electrophoresis. This would be almost like a native gel. So this is the simplest form of CE, where you don't do anything to your sample and you're just separating your sample by charge to mass ratio. You can do capillary gel electrophoresis, so this is also called CGE, or it's called CESDS. So separation occurs in a gel, but this time this polyacrylamide gel is inside the capillary. And SDS can be used to coat your protein in negative charge, so you only get separation by size. So same as in SDS page. Then you can use capillary isoelectric focusing, where you are separating sample based on charge that occurs along a pH gradient. So protein will migrate depending on their isoelectric point. So remember, isoelectric point is that pH of your sample at which your sample is neutral. So pH at which your sample does not have any charge. So your capillary zone electrophoresis, or CZE, as I said, that's the simplest form, and separation is based on differences in the charge to mass ratio of analytes. So in this case we use unmodified fused silica to separate charge analytes, because we do want isoelectric flow. So we don't want to suppress or we don't want to neutralize EOF. EOF is actually helping us to separate our sample. So the way it works is, so everything that you inject into your buffer will flow with EOF towards the cathode, but separation will occur because your analytes will have different electrophoretic mobilities. So things that are going to move quickest are going to be cations with the largest charge to mass ratio. So those ones that are kind of the smallest and have the most positive charge. They would be followed, but then with cations with reduced ratio. Then you would get your neutral species that don't really want to move but are moving because of EOF. And then you get your anions with their negatively charged and don't really want to go towards the cathode but will be swept because of EOF. So first you get anions with smaller charge to mass ratio, and then the last ones that will come through your detector will be anions with greater ratio. So EOF is really kind of helping you to separate these anions and cations. Capillary gel electrophoresis. So this is just an adaptation of your traditional gel electrophoresis, but now you're using polymers in solution and you're putting it into capillary to create molecular sieve. You add SDS and boil your sample, so that negatively charges all of your proteins. So everything will move towards the anode this time, so positively charged electrode, and separation is going to be only by size because we can ignore charge because we put overwhelming amount of negatively charged, so everything has to move towards the anode. And larger molecules are retarded more by the viscous separation medium than smaller molecules. So smaller molecules reach the texture faster than larger molecules. Media that we use for CGE is slightly different than the polymers that we use for gels. So polymers that we use for gel, slab gel electrophoresis, were used traditionally for CE. They were agarose and cross-linked polyacrylamide, and they were prepared directly inside the capillary columns. So you would polymerize it in your capillary column. This led to a lot of issues because when you are doing something like this, you're polymerizing your network of your branches inside the capillary, they sometimes shrink and break during polymerization, and then you get bubble formation inside the column. So if you have bubbles inside the column, that will really not help with your sample traveling through it. So it will stop your sample traveling through the capillary. And then over time, large molecules and different particulates were accumulating at the end of the column, and they would clog the column. So you couldn't really use this column for a long time. So once you made it, you couldn't use it for a lot of injections because it would just get blocked with all the particulates. And so run-to-run reproducibility was extremely low. So now the medium that is used to make gels inside the capillary are more kind of linear and branched polymers that don't form as strong bonds of strong network as these network polymers that were used before. So currently water-soluble, replaceable linear or slightly branched polymers are used. So things like linear polyacrylamide or linear polyethylene glycol or dextran. So they are you dissolve them in a buffer and then you hydrodynamically load it onto a capillary. And then with each injection you can actually like flush them out and put a new gel in. So you don't get uh, hopefully bubbles if you are careful, but you don't get that blockages. So you don't get particulate being accumulated because you can replace your medium, your separation medium with each injection. But capillary wall for this method needs to be coated to eliminate EOF. So in this case, we don't want EOF. We don't want EOF to separate our samples. We just want our samples to separate based on size because this is, SDS separation. So we just want based on size. We don't want EOF. So EOF needs to be eliminated, usually by adding something like acid to protonate those silanol groups, and also these branched linear polymers, they also help to neutralize EOF. So your capillary gel electrophoresis can be used with UV or it can be used with laser-induced fluorescence, and if you need to increase sensitivity, you will use laser-induced fluorescence. And then you can see your separation. For example, what you can do is, if you remember, so we are adding SDS, but we can also add reducing agents, so things like DTT or beta-mercaptoethanol, which are going to break disulfide bonds, and you can see the difference kind of more in details what is happening with your sample when you do this. So if we analyze non-reduced monoclonal antibody, so remember that's the monoclonal antibody in which disulfide bonds are not broken, what we see is our main peak is just our intact antibodies. But if we, for example, have any kind of impurities, we will see little impurities here. Okay, so this one is pretty pure, so we don't see impurities. We are only seeing intact antibody as we are expecting. But if we got any degradation of our product, we would see it here as little peaks. If you look at reduced monoclonal antibodies, so in this case we broke the sulfide bonds, so we break our antibody into heavy chains and light chains. So what we see here is main peak of our heavy chain. Then we have our light chain, so you can see light chain is smaller, so it came out first, heavy chain came out later. But we can also see a little peak of non-glycosylated heavy chain. So there is always a part of your antibody that loses sugars. You don't want to have a lot of those because sugars are usually really important. So you can kind of look at it and make sure that you don't have too many. So Manuel is asking, would you be able to see isoforms? Yeah, you can see different isoforms, so we will be mentioning that. We can see charge, different kind of charge variants. We can see, you can also see things like if oxidation happens. So yeah, it's a really good method, really good orthogonal method to your HPLC to confirm different isoforms that you're seeing. Okay. And then, yeah, another one that is really important separation method that is used is capillary isoelectric focusing. So this is used for charge heterogeneity, and it's also good to determine identity, purity, and then post-translational modifications. Like if you have some proteins that became deamidated or phosphorylated, or different kind of sugars on them, and it's good for stability. And in this case, we are separating our sample by their isoelectric point in a pH gradient. And in this case, also, we need to minimize EOF. So we don't want, once we form our pH gradient inside the capillary, we don't want EOF to move everything around. So EOF needs to be minimized with this technique as well. So the way this works is you fill your capillary with amphalites that actually establish your pH gradient. So amphalites are just a mixture of large number of amphoteric species that contain both ionizable acidic and basic functional groups, and they just establish a linear pH gradient inside your capillary. So pH gradient that can be, for example, from pH 3 to pH 10, or maybe pH 5 to pH 8, depending on what you need. You also have your PI markers in there that bracket the target protein as close as possible. So those are little peptides that they have certain PI which where they should be. So they're kind of like a control for you to bracket, and you know where certain pH is. And then you pre-mix all the different solutions and you fill them into the capillary. So in your capillary you have your carrier amphalites that will make your pH gradient. You put your sample proteins, you put your standards, so things like PI markers. They're almost like molecular weight markers. We discussed them as well. So something that has a known PI, so that you can help it to control. You add polymers to help you neutralize EOF, and then maybe some other additives like urea to stop your protein from aggregating, for example. So everything is added into a capillary. And then one end of the capillary is connected with the low pH electrolyte or analyte, and the other end of the capillary is connected with the high pH electrolyte or catalyte. So something like maybe phosphoric acid, and then you have sodium hydroxide. And then you apply electric field. So now you have everything, and you apply electric field. So hydronium ions from the analyte will move towards the cathode, and hydroxide ions will move from the catalyte will move towards the anode. And as they are moving, they will start sorting all of your proteins and your amphalites. So everything is in there. So as all of this is moving, everything is kind of started to move. Your pH gradient will sort itself from pH 3, so 3, 4, 5, 6, 7 to pH 10, for example. And your proteins will also start sorting themselves. So they will just start traveling inside the capillary. They will travel either towards the anode or the cathode, depending on their charge. And they will travel to that pH gradient and they will gradually change their charge as they're traveling to pH gradient and the gradient, and once they come to pH gradient at which they are neutral, they will stop. So that is calling focusing. Okay, so at the beginning of the focusing process we have a high current because everything is moving, but this current is then decreasing over time as our samples and everything is reaching their PI points where they're neutral and they don't move in electric field anymore. Now we don't have anything to transfer the current, so we'll get a real decrease of current, usually 10% of initial value. Okay, so proteins are focused now and they don't really move anymore. So they focus at certain part of pH gradient. They don't move, but how can we detect them if they are focused, for example, at pH 3 or pH 7, and detector is at the end of capillary. So to be able to detect them, what we need to do after focusing is something that we call mobilization. So to detect analytes, then we need to move them towards the detection window. And there are two approaches to do that. We can do this by hydrodynamic or pressure mobilization, or we can do it by chemical mobilization. So basically pressure mobilization, so once everything is focused to their PI point, we can apply slight pressure to your analyte vial, and then all the content will push forward and every zone is driven to the point of detection. So everything will be pushed and then we can detect all of it as they are pushed and being detected. But you do get some broadening of the zones because now you are moving into that laminar flow a little bit more because you're pushing something rather than applying electric field. So you can lose a bit on resolution, but it's a good method. Or you can use chemical mobilization. So you basically replace your analyte or your catalyte with some other electrolyte solution with different pH or ionic strength. So for example, sodium hydroxide can be changed to sodium chloride, and then this will disrupt the pH gradient and it will cause analytes to acquire charge again, and they will start moving towards the outlet. And those ones that are closest to detector will of course reach the detector first, and we will detect them first on our electropherogram. Okay, so Rita is asking pressure pump use. It's not really the pump use, you're just pressurizing your reservoir to push something through. Rita is what pressure range of that really depends on the system, and it really depends on the method, so I wouldn't be able to give you like one number, but I can show you when you are in NIBRT, for example, some of the examples of some of our methods. But as I said, they would really vary on the system that you have. Okay. And the sample, of course, because some samples are a bit more sensitive than the others. But of course, yeah, I'll show you some of our ones. Okay. So this would be chemical mobilization. Okay, and that's how so basically we're just disrupting that pH gradient, making everything charged again, and then because things are charged, they just start moving inside the fluid. So there are some issues with this mobilization process. So as I said, sometimes you get distortion of the linear pH gradient and you get some deterioration of resolution. Mobilization will take a long time. So it might mean that, maybe just an hour out of your method is just its mobilization. And sometimes, well, lots of times it's really difficult to determine where to stop focusing and where to start mobilizing. So how do you know that everything focused and now you can mobilize it? If you don't wait long enough, samples are not gonna focus. They're still gonna be traveling and trying to find their PI. If you are moving them, if you wait too long, your samples will start aggregating and blocking the column. So it's kind of hard to figure out when is the best. And it's quite a lot poor reproducibility for this method. So it's different for different sample and really depends on operator's experience. So to kind of fight these disadvantages, there is now new system on the market for imaging capillary isoelectric focusing that is being used by a lot of QC labs now. So it was developed, a kind of idea came from 1990s from Wu and Pavzin, And they introduced whole capillary imaging detection to avoid necessity to move samples beyond the point of detection. So they actually made the whole capillary detection window. And then this approach has been commercialized by a company called Protein Simple, and they made an instrument that was called ICE 280, and now it's called ICE 3, and it's being used a lot. So basically what you have here is your whole capillary is a detection window. Your UV light source is shining on through the whole capillary and detecting everything in real time, and you have a camera that is taking images of your capillary. So a CCD camera takes UV absorption image of the entire capillary every 30 seconds. So it's actually allowing your real-time monitoring. You can see at real time when things are focusing, and you don't have to move anything to detection window. You're actually detecting it and seeing it as it's happening inside the capillary. So much easier to do because you can skip that mobilization process. And then your separation pattern is captured and analyzed. So basically it's seeing something like this. When your sample is loading, everything is kind of heterogeneous, and then as it's focusing, you can kind of see samples traveling, and then you can quantitate it and get your peaks and analyze your samples on your protein. How long does it take for one sample to run? So on your ICE instrument, samples usually take from 8 to 12 minutes. Kind of depends on the method, compared to a normal one that would be over an hour, hour and a half, maybe longer. Okay, so much quicker with ICE. So companies that use isoelectric focusing a lot, it's definitely worth for them investing in a separate instrument like this. So the instrument that I mentioned, PA800, that can be used to do, you know, your CZE, your CGE, your isoelectric focusing. This instrument can only be used for isoelectric focusing, but it's just it's much more reproducible, much easier to use. It's definitely worth for companies investing if they have a need for it. Okay, so compared to gel electrophoresis, capillary electrophoresis is much more automatic, giving you much higher resolution. You have this online detection, which means it's quantitative. So you have the detector online that is measuring your UV, and you really need a little of, you don't need a lot of samples. So you're basically injecting nanoliters of samples. You do need to put microliters of samples so it can be injected, but you're not using all of that sample. One disadvantage is you don't have that parallel operation. So you're basically you can't run all of your samples at the same time like on a gel. So every sample is a new injection. So everything is a new injection. Okay, so even though it is quicker compared to running a gel, it still takes time because you need to inject every sample separately. Okay. Capillary electrophoresis can in some cases be coupled with mass spec. Usually it's not really compatible, and it's still kind of, this is still in development, but just so you know that this is probably the future, so it would be amazing if you can couple it with mass spec. Problem is that we are using buffers, charge buffers, which then are kind of interfering with mass specs, but more and more companies are looking into this to be able to connect your capillary and then proceed after purification with your capillary to proceed to mass spec and kind of analyze your samples a bit more. Okay, so application in biopharma for CE. So you can use it to determine molecular weight of your protein. So, you know, doing your SDS, using your molecular weight markers, generating standard curve, and then determining a known molecular weight of your unknown protein. You can use it for IgG purity and heterogeneity determination. So I'm going to show you that. You can use it to look at charge heterogeneity, so capillary isoelectric focusing. You can analyze carbohydrates or glycans with C-LIF, so you can make them fluorescent and then analyze them using laser-induced fluorescence. And it's used quite a lot for small molecular drug analysis and ion analysis. So the determination of molecular weight, so you can denature your protein with SDS. You separate it based on size. Of course, as we said, smaller molecules move faster than larger. and then you can determine apparent molecular weight of the sample by creating a standard curve of known molecular weights. So, for example, you inject all of your known molecular weights, all of your standards. You can see what their migration times are. So, for example, for 20 kilodaltons would be 6 minutes, 35 would be 8 minutes, 50 would be 10 minutes. You can plot that onto a graph, and then you can figure out what is the apparent molecular weight of your unknown by looking at your graph. So in this case would be 11 kilodaltons. Then for purity and heterogeneity determination, so this allows you resolution IgG isoforms and impurities, and you can analyze it in non-reduced and reduced forms. So remember, if we have non-reduced IgG, so if we didn't break disulfide bonds between heavy chains and light chains, what we are expecting to see is your intact tetramer, so your intact antibody, and most of it is going to be glycosylated, but you might have some percentage of antibodies that lost sugar, so some percentage of non-glycosylated, so you can see whether that meets your specifications or not. If you reduce it, so if you break disulfide bonds and you break this into 4 chains, you are expecting to see 2 light chains, you're expecting to see 2 heavy chains, and those heavy chains can be glycosylated or non-glycosylated. So basically with the pure monoclonal antibody, you can see something like this. So in the first case, we have your reduced IgG, and so for pure antibody reduced, remember, we are always seeing light chain, we are seeing heavy chain with sugar mostly, and then you can have a small percentage of non-glycosylated heavy chains. And then usually you have so you spike all of your samples with your 10 kilodalton standard, so you can actually align all of these graphs when you're analyzing it. So this would be, you can see in case you had some impurities or anything like this, you would see more peaks than these 3 that I told you. In the sample underneath, we are looking at non-reduced IgG, and what we are seeing here now is we are seeing mostly our glycosylated IgG. We're seeing a little peak of non-glycosylated, and then you might see some of your samples. Some of your antibody will always break into smaller pieces. So you are allowed to see certain small percentages of antibody that lost 1 light chain, or antibodies that only has heavy chain, and then you see little light chains to get light chains here, because antibody does get a little bit broken up in your process. But as long as this is really low and within your specification, you're happy. But just to show you how kind of more in depth you can go into looking at what is actually happening with your antibody in your sample. So this is not really something that you would be able to see just on a gel. And then determination of isoelectric point. Sorry, Rita is asking, can you quantify protein from peak area? Yeah, of course you can quantify them from peak areas. So again, you have, you determine your software depending on your integration. So we'll determine the size of all the peaks together, so that would be 100%, and then you can get quantitation of each peak depending on the peak area of all of them. So you can get percentage of peak area for each peak, and that can be something that is quantitative and that is in your specification, and that you can then compare between samples. So definitely quantitative, same as HPLC, for example. Okay, so determination of protein isoelectric point. Okay, again, you can inject something of known PI, and then you can make calibration curve. So you use standards with proteins of PIs that are known, and plot mobilization time versus PI within defined pH range, and then you can determine the pH of your protein. So for this one, this is a complex monoclonal antibody, and isoelectric points of this antibody, you can see that we have quite a lot of peaks, so quite a lot of charge isoforms, and these isoelectric points of them varies from 6.73 to 7.46, and this is estimated using a standard curve, just like with everything else. Okay, and then you can analyze charge variance by CIEF. So again, this is just a comparison of originator and biosimilar, and we are looking at different isoforms. So you can see we have the main peak here, the main isoform that has pi of 8.7, so that's the main molecule, but there is some isoforms that are going to be a bit more acidic and a bit more basic. So a bit more basic are on this side because we have pH 9 on this side, and then we have pH pi 8.2 on this side. So we can compare them, and you can see as long as we are kind of seeing similar peaks, we know that everything is fine. Okay, so this would be it for capillary electrophoresis. So just to summarize, we talked about principles of C, so separation of analytes in a narrow capillary under influence of electric field. There are different types, a capillary zone, capillary gel electrophoresis, and capillary isoelectric focusing, and it's used to look at size, charge, purity, stability, and heterogeneity of our sample. Okay, so do you guys have any questions about this? Okay, so next week we don't have a lecture because it's you have your semester break, so no lecture next week, and then we will be back with lectures in 2 weeks. Okay, someone is asking. Tina is asking regarding the test, could I provide with correct answers to compare? Okay, so yeah, so if you check tomorrow, I didn't open the option to see correct answers because not all of the students did the MCQ, but now all of you guys did your MCQ, so I'm gonna open that option so you can see your results, and if you check tomorrow, you will see your results. Average score for the group, you guys cannot see average score for the groups, you can only see your own results. Okay. So even this evening, I'll try to open it now, so you should be able to see like what you answered wrong and what was the correct answer. Then Manuel is saying, question about project is meant to be between 4,000 6,000 words. Is it with abstract and references? So yeah, 4,000 to 6,000 words. Well, it doesn't really matter. It can be with abstract and references. It can be just the main part of the body. It's just kind of like a guideline, so you don't have to worry too much if it goes over a bit because of the abstract and references. Mostly that the main body is between 4,000 and 6,000. Always asking what reference style do we have to use? Yeah, Harvard reference style is the one that we prefer. If you think of any other questions, just email me and I'll see you guys in 2 weeks. Bye.